Great. Hello, everyone. I'm Lakshmi Subramanian. I'm a program manager here at Seven Bridges, and it's a pleasure for me to welcome you all to the fourth month webinar of the CGC Monthly Webinar Series. So I would like to thank all of you for continuing to support our webinar series and also for using the CGC. Just a few announcements, information about upcoming webinars, slides and recordings will be available in the CGC website. You can go to the cancergenomicscloud.org/webinars. You'll find all the details. As by now you're probably familiar, these webinars happen on the fourth Wednesday of each month at 2 p.m. Eastern. And if you have any questions, please use the chat box for asking the questions. At the end of the presentation, we will try to answer as many as possible. I will post a link for a survey at the end of the seminar. So please respond to the survey. So that'll help us with improving the webinars and also, doing more topics that you all are interested in. With those announcements, I would like to introduce the speakers of today's webinar, Dr. Vesna Pajic and Nevena Vukojicic. Dr. Pajic holds a PhD in computer science. She's been working as an associate professor at the University of Belgrade, teaching several courses in mathematics and computer science. Her research topics were related mainly to the application of mathematics in life sciences. So she developed interest, obviously, in bioinformatics, and she moonlighted as a bioinformatician. She's been a member of the Seven Bridges bioinformatics team for more than 4 years, and she's currently working as a team lead, managing over 5 bioinformaticians. Nevena is a bioinformatics analyst at Seven Bridges. She holds an MSc in fungal biology from the Faculty of Biology, University of Belgrade, where she's currently enrolled in PhD studies. So prior to joining Seven Bridges, Nevena worked as a research trainee at the Faculty of Biology, Department of Biochemistry and Molecular Biology. Her focus was on the biochemical and molecular basis of host pathogen interactions. So today, Vesna and Nevena will describe various features of CGC that enable multi-omics analysis, particularly in a use case involving breast cancer. So they're going to be combining genomics, non-coding transcriptomics, and proteomics. So that's really exciting. So we're moving a step ahead and doing true multi-omics. I'm going to show you how it can be performed on the CGC. So without much ado, I would hand over the floor to Vesna.
Okay, thank you, Sai. I hope you see my screen now.
Yes.
Yeah, great. Yes, so thank you and welcome you all to our presentation. During our first webinar in 2021, Dave and Sai gave a nice high-level overview and a live demo of Cancer Genomics Cloud and its features. And today we will follow up on that and present to you in detail how some of these features can be used for integrative analysis of data coming from different omics experiments, which is commonly referred to as multi-omics analysis. So for those of you who are maybe new to CGC, I suggest to check this January webinar for more information on CGC platform. Today, I will start from short introduction to what CGC is and what type of resources you may find on it, focusing on multi-omics resources. Then I will go through different aspects of multi-omics analysis, such as levels of data integration and the methods that can be used for analyzing such integrated data. And the next important thing would be to see what features CGC has to offer that can be used in multi-omics types of analysis. In the second part of today's talk, Nevena is going to walk you through an analysis of breast cancer data from TCGA and CPTAC and show you on a real example how you can utilize those CGC key features for your own research, from searching for data to joint data analysis. The project she will use for this demo is going to be publicly available on CGC, so you can try everything Nevena will present today. And at the end, I will tell you about 2 ongoing projects we currently work on and our future plans regarding multi-omics. Okay, so let's start first with a short overview of resources on CGC that can be used for multi-omics analysis. So CGC started as a project in 2014 with a grant 7 Bridges received from NIH. And within this project, 7 Bridges have to develop a cloud resource that can be used within NCI Cancer Research Data Commons. And although it should have included multi-omics data sets from the start, it was named Cancer Genomics Cloud at the beginning. So from this initial project to date, CGC keeps progressing and evolving constantly. And today, we see CGC as a cloud-based environment for collaborative research and bioinformatics data analysis. So what does it mean exactly? Cloud-based would mean that everything is stored and computed on the cloud. And from the user perspective, it means that it is accessible from anywhere and you will have full cost and access control. Environment would mean that it is not like one web application or a program, but it is rather a set of services, web pages, applications, and API packages for managing data and running analysis. It is meant for collaborative research, meaning that you can easily share your projects and your results with your team members in a very secure manner. And it is meant for bioinformatics data analysis. Practically, you can store any data and process any data on CGC, but a majority of its features are customized to omics data file storage and visualization. And for analysis, you can run different apps. These analyses are reproducible and shareable, and they're already ready to use apps on our public apps gallery. And of course, you can create your own apps or workflows. During all of these years, CGC grew not just in terms of these services it has to offer to its users, but it also became a place for sharing data coming from many different omics data sets. You can see here what this progress looks like, and we keep adding new data sets almost quarterly. Some of them are TCGA, CPTAC, data from ICGC data portal, and many more. Within this variety of data sets, you can find data from different experimental setups, such as RNA sequencing, whole genome, 4-exome sequencing, bisulfite sequencing, ATAC-seq, ChIP-seq, proteomics, even imaging. Data are stored in different formats also, from raw, unprocessed data, such as FASTQ files or MZML files, to VCFs or gene expression tables. And in parallel with adding data sets to CGC, our bioinformatics team has been developing tools and workflows for analyzing data. And more than 570 apps can be found on our public apps gallery, with possibility to create new ones or customize the existing. There are workflows for almost any sequencing technology available. So having all these resources, such as data from diverse omics technologies and environment for running a variety of analyses, are we ready to say that CGC is nowadays a multi-omics environment? And maybe we can change its name to something like Cancer Multi-omics Cloud. I would say yes, but let me try to convince you too. So first, before that, let's see what multi-omics analysis stands for. So today, everybody is talking about multi-omics analysis as one promising approach to reveal functionality of complex biological systems and processes. And unfortunately, it is often naively taken as some magic process where you can just put together all the data you have, and then usually by applying some machine learning algorithms or other fancy stuff from computer science, you just end up with some novel knowledge. Well, in reality, it is a quite complex process, and it needs to be very carefully designed and conducted in all of its steps. So from sampling to interpretation of the results. We now do have numerous data sets with data coming from different omics technologies, but not all of them are adequate for multi-omics analysis. And on the other hand, there is a lack of formal methodology. Instead, a number of statistical and machine learning methods have been adapted and used for this kind of analysis, but it is now quite hard to choose the right method without formal methodology framework. So choosing the right method for data integration and for the analysis will be mainly driven by the concrete application and the question or hypothesis which are in focus of research. And here are some of the possible applications listed here. So, for example, you may wish to find disease subtypes based on data modalities coming from different omics experiments, or to identify driver genes of some disease that are specific to a patient. The concrete application will have a unique biological question or research hypothesis you will try to answer or to test within multi-omics experiment. But beside this biological question, there are some technical questions you should also consider when choosing the methods for the analysis, like which types of omics platforms will provide the most value, or how are the samples collected and prepared, or how are the multi-omics data going to be integrated. So all these and probably some more should be taken into account during the design of the analysis. When it comes to integration of data, it can happen on different levels and in different steps of analysis. So in literature, we may find a different classification of data integration approaches, such as vertical and horizontal data integration or multi-staged and meta-dimensional integration models. Vertical integration, for example, refers to the integration of omics data from different experiments on same samples, while horizontal refers to integration of same set of variables across different studies. In another paper, these approaches are called multi-staged and meta-dimensional. So if we look into the point within the analysis flow, when this integration occurs, we may classify data integration approaches into pre-secondary, post-secondary, and post-tertiary. Pre-secondary integration happens on raw files level. For example, if you have FASTQ files from single-cell experiments, such as single-cell RNA or single-cell ATAC-seq, and you wish to mimic bulk RNA or bulk ATAC-seq by joining reads together. Post-secondary integration is done after the secondary analysis. For example, you can join together raw counts from gene expression analysis and protein intensities from proteomics experiments. This is what the authors call vertical integration. And post-tertiary is integration of results from tertiary analysis, such when, for example, differentially expressed genes and protein biomarkers have been detected individually, and the integration is done at the end. So this kind of integration is usually used for confirming results obtained with one technology based on the results from another. So after you choose your data integration approach, you need to see how you will analyze them. So there are numerous methods and code packages available, coming mostly from the field of statistics and machine learning, which have been adapted and used for some specific tasks within multi-omics analysis. Here is one nice graph. It is taken from a review paper about multi-omics data integration from 2020. And you can see on it some of the methods developed in recent years, grouped by the methodology they're based on. So, for example, methods based on Bayesian approach are statistical methods, which can be used to derive probability distributions of sets of unknown parameters, modeling not only their values, but also the uncertainty associated with it. And such methods have wide applicability. And in this context, they are used for all three types of applications presented here. So disease subtyping, disease insights, and biomarker prediction. And if you're not an expert in statistics and programming, you shouldn't be demotivated to do multi-omics analysis. Most of these tools presented here and other packages also that you can find offer very nice tutorials and examples of analysis that can be easily reproduced with a minimal technical knowledge. And one of these packages, MixOmics, will be presented today during the second part of this webinar, and you will see how you can use it directly on CGC platform within your project. When you are done with designing your analysis, during the implementation, you will probably face different challenges. Some of them are presented here on the left, and on the right, you can see some techniques that you will probably use to overcome these challenges. So, for example, individual omics data are very heterogeneous, and they are usually generated using wide range of platforms. They have different formats. So you will probably try to unify them and prior to analysis with some reformatting or normalization. Then files produced with today's NGS sequencers are large and difficult to transfer, to manage, and to process. So you will need probably some flexible computational resources that can process such files, but which will also allow you to control and optimize your costs. And also, you will need some kind of file management system in which you can easily store your files and share them with your collaborators. Let's see what are the key features of CGC platform that will enable you to implement all of these techniques and ease your analysis and make it a reality. So regardless of a chosen method or application, almost all multi-omics analysis will have a flow similar to the one shown here. You will start from some hypothesis important for your research, and based on that hypothesis, you will design the experiment, collect samples, and process them with different omics technologies. It can be NGS, it can be mass spectrometry, or similar. And you can also use the same omics technology, but on different sample cohorts. Either way, you will end up with raw data files with diverse contents and formats which you wish to analyze jointly. And this is the point where CGC can help you from. So this orange line here rounds all the subsequent tasks that can be done within multi-omics analysis by using CGC platform. So you can process raw files with numerous ready-to-use pipelines. You can prepare resulting data for joint analysis directly with our data cruncher on CGC, and you can also analyze the data there. All these tasks can be done within one project without the need for transferring data, and with all the other options for collaborative work CGC usually offers. So the first of the features I would like to present today is file management system on CGC. It is a system that allows you to store files in projects and to organize them into folders the same way you would do on your local infrastructure, for example. And files can be added to projects from local or cloud storage, or you can also use publicly available files from our public file gallery or from some of the data sets we mentioned at the beginning of the presentation. Each file can be annotated with metadata fields. And these metadata fields can be defined by the CGC platform by default or defined by the user. On the right side, you can see a file page showing its metadata fields. And the information stored in these metadata fields can be used for searching and filtering files on files page. So what is important about CGC file management system? Well, it is not limited to only one type of file. So you can store practically any file type. And the metadata scheme used for annotation is also quite flexible. There is a predefined set of values used by CGC platform, but you can add custom fields, either by CGC graphical interface or by uploading manifest files. It practically means that you can annotate your files in your research-specific way. For example, you can use some phenotypic or clinical data. The other nice thing is that CGC frontend has integrated several file viewers for file types commonly used in different omics experiments. On the left side here, on this slide, you can practically on this slide you can see two pictures, and both pictures are showing a preview tab within file page. On the left, there is BAM file previewed, and Genome Viewer is used for that purpose, and it allows you to see reads and reference tracks side by side. On the right side, TSV file is previewed, and now the tabular view is used, and it allows you to sort files, data based on any column value simply by clicking on column head. The next feature you will probably use for multi-omics analysis is Data Browser. It is an interface for searching and filtering data from public data sets such as TCGA, CPTAC, ICGC, and other. You can create very complex queries to find files for your research, and after you find those files, you can easily import them into your project. Nevena will show you how we did this for breast cancer analysis use case later in this presentation. But I just want to mention here that using Data Browser requires some practice, especially for complex queries. So I just suggest you to check tutorials we have on CGC documentation pages on how to find data for your research and how to use Data Browser. Yeah, sorry. So the next nice feature I would like to present is Data Cruncher. It is an interactive analysis tool which uses Jupyter Lab and RStudio as computational environments. And it allows you to perform analysis directly where your data is. So, for example, if your data sets are stored on Google buckets, the analysis will also run on Google Cloud instances without any additional transfer costs, such as egress. And the outputs are also saved to a CGC project. In multi-omics scenario, Data Cruncher can be used for tasks such as filtering, normalization in pre-processing phase, or for writing joint data analysis, and for preparing custom reports, custom visualizations, and so on. Next, you will probably use apps on CGC platform. An app is a program specifically prepared, so it can be run on the cloud, and it is based on CWL for describing the command line, for its inputs and outputs, and links to other apps, and on Docker technology for describing running environment for that app. It can be simple, just one command line program, and in this case it is called tool, or complex when it is made from several apps connected into one single workflow. During the lifetime of CGC, we have developed and ported a great number of apps that are prepared and ready to use, and they can be found on public apps gallery, and they support majority of analysis commonly used in bioinformatics, such as aligning reads, variant calling, gene expression analysis, SNP calling, CNV and SV detection, metagenomics profiling, and many, many more. So currently, there are 570 of them, but this list is not final. And additionally, a user has an option to modify these apps or to create its own. There is an app editor available on CGC, and it can be used for CWL code modifications. And what is important here in multi-omics scenario is that there are no limitations regarding omics technology. So you can port any type of analysis to CGC. And furthermore, we are currently developing few apps that could run the whole multi-omics analysis as well with a single click. I will talk more about them at the end of this presentation. So after preparing data and picking an app for the analysis, you can easily control how this app is going to be executed. So when you run your app, the draft task page will show up, And there is a part for setting inputs and arguments, but I will not go over it now. Instead, I advise you to check our documentation on this link if you're not familiar with running tasks on CGC. What I would like to do is draw your attention to execution settings part of the task page. Because it contains options to set computational resources and to use features such as memorization in case you want to reuse results from previous runs. These are very important features for multi-omics analysis because they allow you to control resources and costs of your analysis. As you may know, methods used for multi-omics analysis are often very computationally demanding, especially if ran on highly dimensional data. And by setting larger instances for your analysis, you will enable it to finish successfully. And memorization is there just in case something goes wrong and you need to rerun your analysis. In that case, it will save you time and money. Having all that said, Nevena will show you now how CGC is used for multi-omics analysis on a real case. And the projects she's using here will be made publicly available on CGC, so you will have an opportunity to try this analysis on your own.
Yeah, thank you, Vesna. So in this part of the talk, I'm going to walk you through an example of a multi-omics analysis, which we did using the breast cancer data from TCGA and CPTAC, but it can also be done with your data or any other data with some slight modifications. So our first steps were searching for the data and importing the data to the project. So we used breast invasive carcinoma dataset, and we had 99 samples from 3 datasets total, which were proteomics dataset coming from CPTAC and mRNA and miRNA datasets, both coming from TCGA dataset. And as Vesna said, we all have all these data sets available on CGC. So we used the data browser for importing them to the project. This is an example of how the data browser's query looks like. So here we search through CPTAC dataset, and here you can see that we chose the appropriate disease type and file type. Here we chose proteome, and we ended up with 108 files, which we then copied to the project by using this button in the upper right corner of the screen. There is also another way how you can import proteomics data to CGC, and that is through Proteomic Data Commons or PDC. You can actually go to their website. You can make a query for yourself for this same data, for example. Then you can download the file manifest and import this manifest file to the CGC, like this shown at the right corner, at the right part of the screen. And then you'll have this same data in your project listed as well. So this is just another convenient option. This is a data browser's query for mRNA data and miRNA data. It's a bit complicated than the last one. Here we search through TCGA dataset. And as you can see here, we have actually 2 nodes. So we chose the experimental strategy to be RNA-seq for the mRNA data, and the other one was microRNA-seq. As you can see here, we actually chose to import the data which are actually expression quantification data, so counts, process data. And we did that by choosing the workflow type to be FPKM, as you can see here, and the appropriate data type for microRNA too. So actually, this is a very convenient feature because you can skip all the processing of the raw reads in FastQC format and just have the quantification data directly ready for further multi-omics analysis. But of course, if you have your own data in FastQC raw reads, We have a lot of different workflows and tools on CGC that you can use for alignment, quantification, and all the prior processing to get the quantification expression data. And yeah, of course, at the end, we also copied this data to the project in a similar way. And so after importing the data, we needed to process all 3 data sets. And with proteomics data, you can see here how the files looked like in the project. And you can see that for every sample, we had 3 different file formats. So we needed only the raw format here. So we firstly needed to filter only the data in raw format, and then we processed them with MaxQuant. And that is a quantitative proteomics software designed for analyzing mass spectrometric data. And it is also publicly available on CGC. And actually, this is how a one task example of MaxQuant looks like on CGC. Here we can see that we used as input one sample in raw format. We used the reference FASTA file, protein reference, and we used the configuration XML file, and those were the inputs to the task. But more importantly, in the middle of the screen here, you can see that you can actually set a lot of different parameters. For example, you can set what the labeling was in your experiment, what was the reporter ion type, and also some computational stuff like the number of threads, which can be useful for faster execution, for example. And then when the task is completed, you get the output folder, which actually looks like this when opened. It has a list of different output files, and we use this one, evidence.txt, because it had everything we needed, which is identified peptides and the quantification values, which we needed for further analysis. So opposite to the proteomics data, we didn't have much processing to do with mRNA and microRNA data, because as I said, we already imported expression data in FPKM values for mRNA and in reads per million microRNA mapped. But we actually encountered here another problem, so to say. So as you can see, this is how this file looked like in the project. And you can notice that their names are pretty random, and this control data cannot be renamed. So their names, they don't actually match with the sample names. Sample names here would be this under the sample ID metadata field. So these strings. And we also noticed that FPKM files were compressed. So we had to decompress the file, and we wanted to rename the files accordingly so we can prepare them for the integrative analysis and to easily join them later. And we did that by using a custom-made tool that can decompress the file and set the name from using the string from the metadata sample ID field. And I'm telling you this just because this is an example of how to make a simple and useful tool by yourself and do stuff like this on the platform too. So you don't have to, for example, with your own data to download the files, change them and upload them again. You can do stuff like this also on the platform. This was a simple tool with just a few lines of bash code. So you can consult our documentation for help and for tutorial on how to use the tool editor and to make your own custom tools when you have some, so to say, problems like this one. So then, of course, we finished all the pre-processing of all 3 data sets, and now it was time to join them together in multi-omics interactive analysis. The analysis was written in R, in DataCruncher's R Studio, and it is publicly available as R notebook, so it can be changed. The code can be modified for your own files. and it's also available as an HTML file. So this analysis consists of 3 parts. Preparation of all 3 data sets, multi-block discriminant analysis with DIABLO, and prediction on an external test set. And we'll go through it step by step. But I just wanted first to tell you a few sentences about the DIABLO analysis, which is the main part of our analysis. So this is a part of a MixOmics R package, and it's a framework for the integration of multiple data sets in a supervised analysis. What does it actually mean? So we give DIABLO input data, a lot of different multi-omics data sets, filtered and normalized, and we call that training sets. And then it aims to maximize this common information between these data sets and to identify key variables to explain and to classify samples into different classes. For example, here the classes were disease subtypes. And then we give DIABLO another data set, test data set, and then it can predict the class of the samples by using the result from the previous step as a model for prediction. So for a demonstration purpose, in our case, we selected 70 samples that were split into training and test sets with an 8:2 ratio. And the aim of our analysis was to identify highly correlated multi-omics signature discriminating the breast cancer subtypes called basal, HER2, and LUM-A. And now I will show you the analysis, and we'll just go quickly through it. So this is how the analysis looks like. Of course, after loading all the required packages, we prepared proteomics data. We loaded the output files from MaxQuant, as I showed you. We aggregated the data from peptides to proteins. Then we performed normalization and transformation steps that are explained in more detail in CPTAC protein report, if you're interested. And then at the end, we filtered the data sets. We omitted the rows with more than 90% of 0 values. And we join all the samples together in one data frame. We also prepared mRNA data by loading the files with expression values, choosing the same samples that are present in the proteomics data sets. And here, for example, we converted ensemble IDs that were present in these gene expression files to gene symbols so that it could match the proteomics file naming. That was important to us because we wanted to filter only genes whose protein products have been quantified in these selected samples. And we also omitted the rows with more than 90% of 0 values and kept only genes that are present in both data sets. And in similar way, we also prepared miRNA data, loaded the expression values, selected only the column with reads per million microRNA mapped, and it also joined all the samples in one data frame. So we had now all the 3 data sets prepared for the main analysis, which is an analysis with DIABLO. And here we firstly loaded the metadata table containing the information about these 70 samples that we wanted to use. And it contained information, it contained their sample names and the disease subtype they belong to. And then we selected these 70 samples from all 3 data sets and created a training data set containing 80% of samples from all 3 data sets. And we used this training data to integrate multiple data sets with one of the DIABLO functions. And finally, we also plotted the samples using 2 principal components, and we produced some additional plots for visualization. For example, here you can see that we created a scatter plot for individual representation of samples for all 3 blocks, and legend being the different disease subtypes. We also visualized correlation between components from different data sets. And you can observe here that first components from each data sets are highly correlated to each other. And we also created this arrow sample plot representing samples from multiple coordinates. And at the end, we did a prediction on an external test set. So we created the test data set containing 20% of the remaining samples. And we used the predict function from DIABLO to predict class of samples using a result from previous steps as a model. And we displayed it here with the confusion table showing a vector of true classes and a vector of predicted classes. For example, you can see here one of the subtypes, basal. So 6 samples were predicted as basal and none of them predicted as the other 2 subtypes and so on. So this was just a quick overview of the analysis without going into code. But of course, as we said, the analysis is publicly available, so you can go through it in details or even try it with our data or with your own data. Of course, if you have any question, don't hesitate to contact us anytime and for anything. And that would be all from me for now. Thanks.
Okay, thanks, Nevena. At the end, here are some of our ongoing projects and future plans regarding multi-omics capabilities on CGC. So one of our current projects is called Multi-omics and Imaging Machine Learning Analysis on TCGA. And the overall aim of this project is to build a predictive model from genomics and histopathological images features in combination with the available clinical data. The model will be used for predicting therapy response among patients with cancer. And we believe that the prediction of mutation based on histopathological images together with these genomic markers, probably by using some deep learning, may have a positive influence on the diagnosis and treatment of patients with cancer, given the importance of gene mutations. The expected deliverables of this project are data crunch of interactive analysis, tools and workflows for deep learning models adapted for imaging data. And all these deliverables will be also publicly available on CGC soon. The other project is called Epigenetic Mechanisms for Transcription Regulation in Cancer. It aims to enable secondary analysis for a few epigenetic experiments, such as ATAC-seq, ChIP-seq, and DNA methylation experiments, and also to integrate results from these single omics experiments in order to characterize epigenetic markers in cancer. Again, all the tools, workflows, and analyses created during this project will be also made publicly available on CGC. And for the future, we plan to continue developing solutions for multi-omics analysis. So there will be more tools, workflows, and data set which you can use on CGC. And we will also implement several new visualization tools and features that could give us better insight into results and help us understand the findings. As for the community-related activities, we will continue presenting multi-omics projects here on CGC webinar also, so you can stay with us if you are interested in this topic. And finally, here are some information how you can ask for help and support on CGC. So you can get basic support for any issue you may have on CGC or any question by simply clicking on the question mark icon, which is present at the bottom right corner at each page on CGC. If you do that, such form will pop up and you can fill it. And by filling it, you will automatically, the system will automatically send your message together with the information about the context the message is sent from to our support team. And that is why this is a preferred way of asking for help. You can also send a message directly to our support team to cgc@7bridges.com. There is also a comprehensive CGC Knowledge Center on this link, and it covers almost all topics you may be interested in with a lot of nice tutorials, so you may want to check it also. And at the end, there is an option to apply for a grant for collaboration on CGC, in which case you can get more advanced support and you can work together with our team in order to design, implement, and run the analysis. On this link here, you can find information about how to apply to these grants, but also there is, I saw there was a question regarding, the question was something like, is CGC publicly available? And here you can find information about that also. The answer is yes, it is. So you can create your account on CGC, and by creating an account, you will get $300 as some initial funds for your pilot funds for your research, so you can try some analysis and store some data. But as I said, if you need more, you can consider applying for this grant here also. So that would be it from our side. If there are more questions, we are here to answer.
Great. Thanks so much, Vesna and Nevena. This was an excellent presentation describing the multi-omics analysis. Yes, we do have a few questions. I know you started with the CGC question, whether it's publicly available. I just want to add another point about that. Yes, it is. You can create an account, and accessing the CGC is available free of charge, but you will certainly need to pay for storage costs on the cloud and also for analysis on the cloud. So to help you get started, we give free credits. The NCI has provided $300 of credits for each user who signs up for an account. So you will be able to look at what is available and then test it out on some of your own files or the publicly available files, and explore the CGC using those pilot funds. And like Nevena mentioned, there are options to apply for collaborative projects that we highly encourage students or PhD students or postdocs to apply for those to support your research work. The other question I saw was, what are the considerations of bioethics and data privacy, considering that this is a cloud-based computational service? So that's an excellent question. There's an entire section on policies in the Cancer Genomics Cloud website. So I would highly encourage you to look at that. Your data is accessible only to you. So that's one of the key features where you control who can see the files you have in your own project. So the project is your own workspace and no one else can see it. But if you choose to share it with collaborators or others, you can actually add them to your project. So data is highly secure in the CGC platform. And we have a lot of security policies. At this point, we are what is called as FISMA moderate, and we are also getting security certifications from FedRAMP. So there are a lot of security policies in place. So next question is there any size limitation in DIABLO in the cloud in terms of variables, columns, observations, rows, or data matrices? Is it limited by the RAM in my local computation environment? Vesna or Nevena, do you want to take that?
Well, yeah, I mean, I'm not sure if that's really a question for us, but yeah, I think it is limited. But yeah, I mean, I'm definitely sure it's limited.
Yeah, so if I may add, it is not limited with your local resources, but I think it would be dependent on the RAM memory available on the cloud instance you are going to run the analysis on.
And it's not just for DIABLO, it's for any other interaction.
Yeah.
It's the same thing, yeah.
Okay, so and the attendee has responded saying, thanks, I did not get that all computations are in the cloud. Yes, so all the computations happen on the cloud. There is another question. Does CGC offer any internships and trainings for grad students? If you do not, do you plan to offer it in the future? So, and I can probably answer that. We do offer tutorials and hands-on training programs for anyone who want to use the CGC. Particularly if you want to host training sessions for graduate students in a university, CGC is a very good tool for training purposes. So we have done such trainings at several universities like Georgetown and others. So if you want to reach us, you can contact us by email. We can provide some more details. But we do not offer any internships, but we can utilize CGC as a training platform for bioinformatics analysis. Any other questions from others? Please feel free to post in the chat. So I had one question, which I wanted to check with Vesna and Nevena while we wait for others. So this whole analysis, you mentioned it will be publicly available in the CGC. So where can the users find this?
Yes, so it will be available within one project. So we are going to publish the whole project. So there will be like these MaxQuant tasks and this tool Nevena has demonstrated. It is called Decompress and Rename.
Right.
And so everything she presented today will be within this project, and the project will be made publicly available in the next few days.
Great.
And it can be found within this menu, public projects. You click on it and you will find it there.
Great. Perfect. Yeah. So they can find it in the public projects menu,
yes.
right?
Yes.
Okay, great. Sounds very good. Does anyone else have any questions for Nevena or Vesna about the multi-omics analysis? Okay. Can you move on to the next slide? I'll just announce the speaker for the next month. Great.
Yes.
So like I said earlier, thanks so much for supporting the CGC Webinar Series. Next month in May, we have Dr. Wenming Zhao, who's a lead bioinformatics scientist at the Food and Drug Administration, FDA. So Dr. Zhao has been a collaborative project recipient and he has performed a lot of analysis on the CGC. So the topic of his presentation is going to be towards best practice in cancer mutation detection with whole genome and whole exome sequencing. So he used the CGC to perform a systematic interrogation of somatic mutations in paired tumor normal cell lines. And he has identified what are the factors which affect reproducibility or detection of somatic mutations and the accuracy of these detections. So on 26th of May, he will be presenting his work where he evaluated the reproducibility of whole genome and whole exome sequencing, testing different sample types with varying input amounts and tumor purity, how multiple library construction protocols affect tumor mutation detection. So please join us for this. The announcement page will be up in the CGC website very soon. The recording of this presentation and the slides will be made available in the CGC website. So if there are no other questions, we can break a little early today. And thanks again, everyone, for joining today's webinar.
Thank you.
Thank you all.